I think now we are in live.
Awesome.
So, so we will start right now.
Awesome. Perfect. Well, thanks so much, everyone.
I will.
For sorry.
Yeah, I will start to introduce
Go
you
go.
first.
Okay, sounds good.
Okay, right. So Abdullah make the option so all attendees can press the lobby automatically.
Okay.
So hello everyone. I would like to welcome to all of you in a new MGBS lecture, and I am extremely happy to welcome to Dr. Micheal Kantar, University of Hawaii in USA, and he will present his outstanding research with title Understanding Plant Germplasm Collections, Data Sharing and Breeding for Climate Change. I would like to thank you, Dr. Mikey,
Thank you.
Dr. Michael, for to show up on time, and please start your presentation.
Awesome. Well, thanks so much for inviting me. It was so much fun meeting you at the conference in Germany, and thank you everyone for coming online. I hope you enjoy it. Well, so I am going to talk about germplasm collections and some ways of how we can use them. But we have to start off a little bit a little bit backwards. Everybody eats, right? This is something that is obvious, but something that might be a little bit less obvious is that while everybody eats, every culture has different favorite plants. Depending on where you are in the world, you have a different favorite plant, and you want to use it for different things. And there's been lots and lots of work over the years on where plants originated, where they came from, and where and how we can best share them. This is a great map that was produced by Colin Curry several years ago now, showing work over the many parts of the world where all of these different crops that we eat are from. And then it went on to show how these crops have traveled around the world and how they've become these major sources of food for everyone across the world. But this is just really a starting point. Everybody eats. We have different favorite food plants all across the world, but where are they stored? So it turns out we've done a lot of work globally and nationally at conserving all of these food plants that we eat. So this great paper by Peterson, talks about how many accessions are held in global gene banks, and we look at some of our major staples like wheat, rice, barley, maize, and we have hundreds and hundreds of thousands of different varieties. And that's really important. Most of these are, well, not most, but many of them are accessible. Hopefully, and if anyone has questions, we can talk about it a little bit later, but there's some great international treaties, the Plant Genetic Resources Treaty, on sharing germplasm globally. But overall, in terms of what we have, there's over 7 million accessions of different crops that people need. But why should you care, other than it's really fun to have all of these collections of plants that are so culturally important? Well, climate change is one reason. So what you're seeing here is a map that was produced by Camila Mora about 10 years ago, where the redder the map gets, the more we're seeing cumulative climate hazards. And the thing that I see when I see this is most of the world is going to be dealing with multiple different climate hazards. Now, there's issues with the way that this map was produced. It's an additive index. We don't look at any interactions, but regardless of that, much of the world is at risk. So one of the ways that we can mitigate this risk is looking at all of this stored variation. There's other reasons as well. So what has happened over the course of domestication and breeding? Well, we have well-known effects of where we have our initial set of genes that exist in the wild. Our wild populations are more diverse than our domesticates, and then through selection, we select for specific variants, and therefore we lose a lot of this diversity. Some of that is intentional because we try to remove deleterious variation, but some of it is just by chance through demographic effects and population size and reduction in those population sizes. But that's why we need to maintain these collections. But because those millions of accessions or hundreds of thousands for specific species, these are the raw material from which we can adapt to these changes, and in particular, unforeseen changes like climate change, where when these collections were initiated 100 years ago, or sometimes longer ago, we didn't necessarily know we were going to be dealing with these abiotic stresses. But this leads to a major question of how do we categorize these collections? Well, there's been a couple of different ways that people have done this over time. The first way was through old school cytogenetics and crossing. So the first characterization were these primary germplasm, which is everything that can be crossed with no meiotic problems. Everything's 100% fertile. The crosses are easy. And the second is the secondary germplasm. So these are typically wild relatives, or even sometimes the progenitor species. And there are some meiotic abnormalities in this case, but you can generally recover fertility and find things that are relatively fertile between them. The next is the tertiary germplasm. Now, the tertiary germplasm is a bit more problematic. In this case, for the crosses, you generally need some size, like embryo rescue, in order to recover plant material, but still, you can do it. It's just more labor intensive. And the last one, which we won't talk about here, but is the quaternary germplasm. So that's any of your genetic engineering or genome engineering techniques, like a CRISPR or something like that. So this initial categorization was really useful and incredibly beneficial, but it is very difficult in many cases when you have dozens, and sometimes many dozens, and in some cases, like with potato, maybe 70 or 80 different species. So this leads to this has led over the last 50 years to thinking more about what are some ways in which we can choose our accessions that are slightly maybe easier, or even within these germplasm categories. Once we figured that out, how do we limit these accessions, right? Because we know a little bit about their meiotic behavior, but we also have hundreds of thousands of choices. So there have been a number of different ways that the folks have done this. One really popular way is FIGS. This is called, so the acronym is FIGS. It stands for Focused Identification of Germplasm Strategy. So this is where you take, for a particular variable, say temperature, you look at the univariate distribution and you pick extreme outliers, right? You pick some outliers that might exist in rare environments, and you say this might be apparent that would be good for that environment. Another method has been the development of core collections. So core collections subset the diversity into a manageable number of crosses, right? And so that's great. A manageable number of crosses is really instead of having 800,000, maybe you have 300. Well, you can work with 300. And in some cases, you can work with more depending on your resources. The next one has been kind of a combination of the Harlan and DeWitt classification of the primary, secondary, and tertiary, along with genetic distance measures. So this has been known as kind of phylorelatives, where we look at the phylogenetics plus those germplasm characterizations. And the last and most recent one is the idea of landscape genomics, where you basically look at the history of how natural selection has worked on wild and landrace populations, and you try and choose either specific loci or specific accessions. Now, all of these methods have different pluses and minuses, but what they all have in common is that they all are trying to answer the question, how can we functionally use this huge amount of diversity, and how can we use techniques from evolutionary biology and applied plant and animal genetics to answer questions surrounding quickly adapting. So let's go through some examples. So let's look at how we choose accessions using FIGS, this focused identification of germplasm strategy. So what you're seeing here in this map is you're seeing species richness for sunflower. So sunflower is native to North America, is domesticated in eastern North America here. And there's lots of species, depending on the taxonomists at any given time. There's somewhere between 49 and 53 different species, all of which are fairly cross-compatible with our domestic annual sunflower. So we can see that these species are all over. So if we take a FIGS approach, in this case, what you're seeing here is we're looking at soil pH and annual precipitation. And what we're seeing in this case is the black box is where annual sunflower is cultivated, and the red box in this case is the phenotypic space that is covered by the primary germplasm. By the primary germplasm. So what we're seeing here is the cultivated space exists in this 500 millimeter, this about 450 to 510 millimeter range, but the primary germplasm exists in ranges all the way from about 250 to about 1,000. So what we do in this case is we take specific accessions that we can pick that are at the extremes of precipitation. And then in this case also soil pH is very important for whether sunflowers will grow, so we can also see if we have variation for that. So that's great, but this is a little bit of a clunky metric. We're still pretty much just using a heuristic of saying, well, we're eyeballing it, it's a bit extreme. And we also then run into this question of what about, other types of stress? What about biotic stress? What about maybe we're picking something that's a range edge population, so maybe it's not great in general. So it's good, but it's not the most satisfying. We can extend FIGS though. So we can look at, instead now of just looking at North America, we can look at potential future climates. So this is all just using current data, or this is using, historic data from 1970 to 2000. We can extend this by looking at what the future climate might look like. So if we're doing that, we can look at which accessions and which regions of the globe, based on species distributions, are gonna what percentage of the populations are going to exist in climates that 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 the sunflowers are not adapted to. So that's what we're seeing here. This 8% shift from climate change. We're moving outside the range of where sunflower wants to exist. Now, if we're looking globally, it's smaller. If we're looking at North America, it's larger. The reason North America again was pulled out here is because this is where sunflowers originated. We can also look at the space of where crop wild relatives, so in this case, our primary and secondary and tertiary germplasm, is it going to help with the global shift? And the answer globally is, well, not really, but in North America, quite a bit. So again, we're dealing with a geography-based approach. So this provides slightly more information than what we had previously, because now we know for North America, well, now we have a subset of species and a subset of conditions that might work, but for other parts of the world, it probably isn't going to work. And what's really cool about this, we can also extend it if we model the distributions of major pests. So Sclerotinia is a major sunflower pathogen. We can also see that in some cases, we're actually going to see some release from pest pressure because places are no longer going to be suitable. So this is kind of useful, but we're still fairly limited. We're thinking about a single growing season. We've done a little bit better because we've now split the geography. But what happens if we look at a different crop? Is this going to hold? So we can do that. We can look at this work that was led by Dr. Malta Stuker here. And one of the things so what we're seeing here is we're looking at now quarters. So January to March, April, May, June. And then what we're seeing here is the gray histogram here is the number of rice successions. So the over 100,000 rice successions that exist in the IRRI, so the International Rice Research Data Germplasm Bank, and where the conditions were where they were collected from. So what we're seeing is in the black chart here is the present day, of the growing region, in this case in the Philippines, and we're seeing blue what would it look like for 2 degrees warmer, and red for 4 degrees warmer. And what we're seeing here is actually quite a promising story where we're seeing a tremendous number of accessions that will in fact exist, or that were collected in regions that exist in these extreme temperatures. Now, of course, there are problems associated with this, as I'm sure many of you are aware of. We're using our mean daily temperatures here, but for rice, nighttime temperatures are often more important for seed set. But at any rate, we're seeing that we have lots of genetic variability, even in our very extreme scenarios. So again, this is promising. The other thing that this allows us to do is think about the phenology. So how much can we change cropping system or planting date? I mean, how much can we change planting date to maybe not have to change our genetics? So this is, again, we're starting to tease out what accessions might work where and when. So we're getting at our spatiotemporal way of thinking about this, but it's still not very satisfying. We're still basically using a heuristic. We're hoping that we're going to be able to cross this well, and in many cases we have no idea of how adapted this germplasm is to our specific region, or how well it will cross with our market types and things like things like that. So figs is good, but it has a lot of limitations. So what about some of the other methods? Well, what about phylorelatives? So this is some work that comes out of Kew Gardens that was done by Dr. Juan Varel here. And so this is looking at asparagus. So what I really want you guys, what I want everyone to look at is kind of zooming in at the bottom of this phylogenetic tree. And what we're seeing here is we're seeing, we have a ploidy series among our cultivated asparagus. And but what we have here is across the ploidy series, we have individuals that have low genetic, or we have species in this case that have low genetic distance, high crossability, and are at the right ploidy state for what we're dealing with. So in this case, where we have limited genetic diversity within a species, we have what we have is we can highlight the best individual, or we can highlight the best species for potential crossing when you move to a system like asparagus that doesn't have the work done in it that species like sunflower or rice have. So we're dealing with more of a niche crop, a crop with less resources, but we can still target some specific species. This is very promising, but it doesn't necessarily help us with the idea of climate change. So how do we combine figs and phylogenetics? So to do that, there was some work that was done by Dr. Nathan Femia, where the explicit goal was to try and combine this figs approach with phylogenetics. And so to do this, potato was used as a model species. And what was really done is we tried to use the breeder's equation, where we're looking at that response to selection, using, so we conceptualized it as this response to selection with, and we used our intensity, our accuracy, which is our, that correlation, and then the variation that's associated with those measurements. So specifically, what this does then is this will not only and what you're seeing here is basically the climates that are represented by the 72 relatives of commonly used relatives of potato here. So really what we're doing here is, and I apologize for the complexity of this slide, but what we're doing is we're using this, we're using the breeder's equation, where we have our selection intensity, but we're modifying the values specifically to deal with the data that we have. So instead of a selection intensity, we're looking at an applicability score here, where we have basically the proportion of accessions that are from a specific Koppen-Geiger climate class, and that's our applicability. And instead of selection accuracy, we're having a combination of environmental plasticity and environmental history. So what is this? Is basically our plasticity is the correlation of a species or overlap with the future projected climate using looking at a number of future climate projections. And where we have our evolutionary history, so this is our phylogenetics component, where we have the regression coefficient of the life history traits based on a phylogenetic regression of how well this is going to predict crossability. And then what we have is basically, instead of that standard deviation of heritability, what we have is we have the variation associated with our environmental plasticity and our environmental history. So if we're going in a little bit further, we can see that for our selection intensity here, we know where the accessions from each of these species exist and the percentages that they exist in a particular climate. For our environmental plasticity, we're looking at this overlap in environmental space between current and future projections. And we're conducting a phylogenetic regression to look at what is resulting in that crossability there. And then we have our coefficients here for our regression coefficient there. And then we actually have we have the variation that we have in the future climate percent overlap because we use many models. And then here what we all have is we have the variance that's associated with the regression models. So what this does is this gives us a nice priority score, a breeding score that is very similar in form and inspired by the breeder's equation. But again, so this gets us to a little bit of a nicer answer because we can tell you, again, instead of broadly saying North America or saying globe, we can say based on your specific climate, tropical rainforest, tropical monsoon, hot semi-arid, we can tell you what species is likely to be your best bet for breeding. But this is still unsatisfying, because now we've done a better job. We've matched based on crossability and climate the species that will most likely work for your future climate needs. But we're still at this problem of having extreme differences in the number of accessions that we have. So how do we move from this large number of accessions for a specific species that we now know is best for a specific climate and will cross well? How do we move that further? Well, this is where a landscape genomics approach might be helpful. So if we're looking at the way that we choose accessions in a landscape genomics approach, and this is work that was led by Dr. Dian Wong, what we can see is we have a lot of the same information that we had before. So we have where accessions are from. We have, in this case, what we're doing is we're associating, instead of looking for extreme accessions, we're associating we're associating the environmental conditions of the location with a specific marker. So we're identifying QTLs that are associated with that variation. And then what we can do is we can look at, again, we can see, okay, is there co-localization among a number of different environmental traits? And what's the difference between our major and minor alleles in this case? And this provides some very useful information. We know what the specific differences you will get or an expected difference in response to an environmental characteristic if you have one of these alleles. Now, it's built on the assumption that natural selection has selected for these, and so this particular technique only works in wild or landrace species because we have broken the linkage with location in many modern breeding programs. But what's really nice is we can get down to specific, in this case, genes that are in LD with the markers that look to have these specific traits. But this technique runs into a little bit of a problem, because even though we can identify these specific alleles, we're doing it in germplasm that's not necessarily the most relevant. And in some cases, for these genes that are really important, they're almost they're actually fixed in some subpopulations of cultivated rice, and even in the ones where it's not, it's at a fairly high frequency. Now, the upside is that even in this subpopulation of the tropical japonica rice, it means we don't have to go back to the wild species. We can actually use elite material where this already exists, and so that's great. Does it work for other species? And the answer is actually yes. And even in species like that are less domesticated than rice, like this is an example of a working cranberry. What we actually see is it's more useful in less domesticated species. So by less domesticated, I mean the cranberry breeding program that Dr. Neihart leads has basically only had 4 generations of selection since the material was collected from the wild in the late 1880s. Cranberry is a long-lived perennial. So what this means is he actually has specific targets, and he's using, and basically in his breeding material, these alleles are not present in his breeding material, so he can make these directed crosses right away. But, okay, so we have an example of this working really well. We have an example of it providing interesting information, but not necessarily allowing us to move to provide as useful as information as we would have hoped. So how can we maybe extend this a little bit further? So one way that we can extend this a little bit further is trying to combine even more of these. So we tried to combine the phylorelatives and figs. That was great, but that didn't necessarily provide us the that didn't provide us the information really that we wanted. So what happens now if we combine FIGs, core collections, and landscape genomics? So this is some work that's being done by a PhD student in my lab, Quinn Campbell. And so what we're seeing here is work in sorghum, where we're seeing, clustering of similar climates of the Ikersat sorghum collection. And then here's just looking at across a range of environmental variables. We're looking at anything in red here are just outliers. So what we're seeing is, by a straight FIGS approach, we have lots of things that are that are extreme, but how useful is that? So what we actually did, so what Quinn did, is instead of using a GWAS approach, we decided to use a genomic selection approach. So what we did is we used the core collection as our training population to predict what the environmental, what the genomic estimated adaptive value, so a GEAV, which is analogous to a GEBV, would be for the entire core collection. So for the entire collection, using that core collection, the mini core collection, as a training population. And so what we see here is by chromosome, we're seeing the values. So in this case, red values are higher, blue values are lower. In this case, good or bad is going to be dependent on the environmental characteristics of your specific location. But what we're seeing that specific chromosomes have different values. We're seeing different values for different geographic regions. But what's really cool here is this gets us to something that we really haven't gotten to before. We get a specific breeding value for a specific accession for a specific environmental variable. Now, in this case, if we're looking at temperature, we're getting a couple of different PIs. We're getting a couple of different accessions that were originally collected from Uganda, and that are, in this case, they are available to be shared. In this case, we're getting, for a different variable, in this case, soil pH, we're getting actually still 2 accessions from Uganda that are adapted to high pH, but in this case, they're different. So again, what's really cool is we're now seeing these variable, we're getting 2 specific accessions for a specific soil or climate variable, and we can aggregate then this information across all of the accessions. And in this case now, we get a value of how adaptable individual country collections are, what's the value for parents for specific accessions in different collections and in different locations. So this provides 2 types of information. This provides, one, we know what accession we want to use for our specific variable, and where should we go to collect? So you can address collection gaps along with picking your best parents for breeding at the same time. So that's really exciting. So we were really excited about this, and a little bit surprised that it worked really well. So then our question was, well, what about, will it work in another species? So a postdoc in my lab, Dr. Anna McCormick, decided to try it in barley. And but she decided to do it with a little bit of a twist. So this is a landrace collection of barley. You can see it's all across the original range, and but what she did that was so cool is she created subpopulation specific distribution models, and then asked the question, well, if things are more related to one another, if accessions are more related to one another, can we get good predictive value? And then can we maybe get around linkage drag by by subsetting the cultivars to those that are already more likely to be used in the region. And so what we saw there is, yes, you could. And you can still get very high predictive values for things like temperature and for variables like temperature. And what you can do, so what you're seeing here is the global population-specific distribution maps and what the actual values of the of each accession is. So now what we have is we can subset within that distribution of interest, what are the individuals that have the highest breeding values and are most closely related to the individuals that we're actually looking for. So this allows you to plan a crossing scheme in a way that is slightly more beneficial. So really, we're really excited about this combination of FIGs, core collections, and landscape genomics because it seems to be providing a better answer than either of these alone, or than the phylorelatives and FIGs, which didn't get us to the same level of precision with our accuracy. So really, this is a lot of talk, but what's the real goal of this? Well, the real goal is to speed up the breeding process. You know, breeding takes a really, really long time. Pre-breeding can be 1 to 3 generations, evaluation and testing another 3 to 4, late generation testing and 2 to 4. So depending on the species, you know, this can be, you know, 3 to 35 years. You know, landscape genomics can reduce pre-breeding by about 50% because you're reducing the number of crosses you make because you've predefined which parents are likely to be for your trait of interest. Genomic selection and speed breeding can speed up early evaluation, and you can speed up your late generation testing by targeting your target populations of environments in a more directed way, which is just very difficult to do, or which is something that we don't often do. So I'm making this big claim about landscape genomics. So can we do it in a lot of species? Well, the answer is yes. So if we look at the Annex 1 of the Plant Genetic Resources Treaty, what are the number of how many crops of that list of 64 crops can we do this in? And it turns out 26. So 26 of those 64, 40% have all of the data necessary to do these analysis right away. And that is something that I'm really excited about because this is public data. This is data that anyone can download off the internet and just do these analyses right away. So I find that personally really exciting because it's a way that we can have the global community. We put all this effort into these resources and we can use this immediately. Any breeding program that you like, you can do this for. So that to me is really exciting. So for the last bit of my talk, I'm going to talk a little bit about something a little bit different. It's about using collections to develop new crops. So this is something that I'm excited about just because I think it's fun to do domestication. But one of the things that we've seen over time is that different crops exist along a domestication spectrum, and they've taken different amounts of time to domesticate. There's a different number of genes involved, and depending on the genetic inheritance or the genetic architecture of these traits, you're basically going to have different limitations. So what you're seeing here is you're basically limited in your ability to domesticate something based on the most complicated trait. If it's a simple single gene trait, super fast. If it's a highly quantitative trait, it takes a longer time. Now, one thing that's important to remember about this is that there's not, while we talk often about a domestication syndrome, there's multiple different domestication syndromes that different societies have used over time. So one of the things that you're seeing here, so this is an example of Zizania palustris, another North American domesticate. One way that folks have tried to domesticate it is to domesticate it as we did paddy rice, where you lose where you get rid of shattering, you increase plant populations, and you can use normal mechanization and things like that. Now, the second way that people have done it has been that is to use more traditional methods, in the sense that this grows in rivers and lakes historically. So what folks have done is they've actually selected for partial shattering, because because then it reseeds as you harvest, and you're taking advantage again of this natural selection process. So there's lots of different ways of doing this. But one of the things that we've seen is that when we know something about the genetic architecture and we know the traits of interest, we can actually get something that's really close to our domestic. So this is a Bayesian simulation here, where we're looking at how many generations of selection and cycles of selection it takes to get something that is similar to our domestic. And what we're seeing is, actually, even with a wild species, we can get something that's incredibly close. So this first 40 cycles, is a burn-in period, and then selection starts at cycle 40. Within about 10 generations here, and in some cases as few as 4 generations, we're getting something that would actually be profitable to a farmer. Now, of course, depending on what we're doing, in this case, we see these massive decreases in genetic variation, which means we probably need to have multiple breeding programs going on, one to maintain, select very aggressively for our traits of interest. But it's something that is, if we know that genetic architecture, we can make progress super fast towards whatever we want. And so it's something that's really exciting. But part of this, and one of the important, is as we start to develop new crops, or even as we start to incorporate genetic diversity from places all over the world, participatory structures are going to be really, really important. And so this is just an example of how to set up a participatory structure from a paper by Dr. Brian Runk. And basically what this is showing is that plant breeders have lots of technical expertise, but they're not always the best at picking the targets of that selection. And so by having more interactions with farmers, more interactions with other stakeholders, you can actually pick your targets of selection better, so that you're going to have outcomes that where outcomes of selection that are things that people really, really want. So really, this is what I want to leave everyone with. We've developed these tremendous amounts of public resources: genomics data, accession data, georeference data, crossability data, phylogenetics data. And if we combine all of this data together, we can actually pick and know what our selection targets are and make really rapid progress by integrating all of this tech together. But to do it, it's not just us operationalizing the technology. It's having deep conversations with the people who, with the farmers and the general population, about how they want to use these technologies, so that we're doing something that people will really, really like and use, because that's the goal. The stuff that we want to do, we want people to use it. So with that, tons and tons of people have been involved in this project. Huge thank you to the crop tusk, Dr. Nora Castaneda, and Benjamin Killian, but lots of people from all over the world: USDA, NSF, Purdue, and of course the people who are really doing the work in terms of Anna, Quinn, and Nathan. So